and the eggs using insulin as the eggs sample. All right, so as we all know, we need insulin, but the condition with the diabetes, right? So everybody produce it, but diabetics, depending on the type, they will need their insulin. So before bacteria, well, this is a crash course, so no history. Let's just get straight to the procedure. So we need to make insulin. So the first thing that we need to do in genetic engineering, what we're doing, we're taking a gene, right? And remember, a gene is just a specific portion of DNA. So we're taking a gene and put it in a vector. So gene goes into vector, vector goes into host, and the host produce the protein. From the gene. So remember, the gene codes for a protein that will be expressed. So you put the gene into a vector, you put the vector into a host, and the host will express the protein. So how does that work? Where does the gene come from? So step one, you need to isolate the gene. Right? After you isolate the gene, this is the basic step. Isolate the gene, place the gene in a vector. Place the vector in a host. All right, so the vector is a plasmid, typically a plasmid. And as I know, plasmid, where do we find plasmid?
Bacteria are prokaryotes.
Right. So the host is the bacteria. All right, so we need a little diagram now to outline this. So you just isolate the gene, place it in a plasmid in bacteria. Anybody writing? I want to clear the screen.
Sir, you isolate the gene. What is the vector? The vector is the bacteria?
No, the plasmid.
And then you put the plasmid into
bacteria.
Oh, okay.
I'm going to show it now via a diagram. So you have your bacteria. Remember now, the bacteria, it will have the chromosomal DNA, right? But we're not interested in the chromosomal DNA. Inside of the bacteria, you have a circular strand of DNA. That is the plasmid. So the plasmid is DNA. So a circular strand of DNA, that is what we call the plasmid. It is inside of your bacteria. All right. So we have our technique to basically punch holes into the bacteria to get out the plasmid. So we remove plasmid from bacteria. All right. And the plasmid is our vector. So once you isolate your vector, we also need to isolate the insulin gene. So in this case, we isolate the insulin gene from the pancreas. All right. Now this is in the form of mRNA. But what we actually need is DNA, right? The messenger RNA, we convert it to DNA. And the enzymes that does this is called reverse transcriptase. So enzymes that convert RNA into DNA, we call them reverse transcriptase. So the HIV virus of that enzyme, reverse transcriptase. We call the DNA complementary DNA. So if you see cDNA, right, copy DNA are complementary DNA. So you convert the messenger RNA into DNA. You have your plasmid. Now we need to fuse the plasmid with the DNA. I am using this to represent the DNA. All right. And this is my plasmid. Right? So what do we need to do? There's a specific class of enzymes that we use when we're doing genetic engineering. Anybody know the name of the enzyme or the class of enzymes?
Are they restricted enzymes then?
Right, restriction enzymes. This process cannot be done without restriction enzymes. Two types of enzymes. The restriction enzyme, which is for cutting, and DNA ligase, which is for joining. So it's like cut and paste. The restriction enzyme is the scissors, and the DNA ligase is the glue. So what we need to do now is cut the plasmid.
Sir, can you repeat the enzyme, please?
Here. So the general name is restriction enzymes. So we're going to cut out a portion of the plasmid. So there you have it. So this is now recombinant DNA. Reason why I call it recombinant DNA, it's DNA from two different organisms. This was from the bacteria and this is from human. All right. And the reason why it's the vector is what does a vector do? So like mosquito is a vector, right?
The gene.
Right. In this case, right, it is carrying something. So the plasmid is carrying the gene. So the plasmid, even though we call it recombinant DNA at this point, so this is our plasmid vector because it has in the DNA. The isolated plasmid, isolated DNA, cut the plasmid, insert the DNA into the plasmid. Once you do that, you place it back into the bacteria. So the bacteria will have its own DNA, and then we put the plasmid back inside as well. So we have our technique to punch holes into the bacteria and allow it to take up the plasmid. All right. So let me just annotate this. Insert plasmid into bacteria. So before you go into the specific, just remember what is the general, the three main things in genetic engineering what are the four main steps?
You isolate the gene, then you place it in a
Isolate the
vector,
gene, place it in a vector, yeah.
and place the
And
vector
then,
in a
Place
host,
the vector in the host.
right?
and then
And
put the
once,
host.
right, in a growth medium. So the host is the bacteria, right? The bacteria must be able to replicate itself. So in order to do that, we're going to now put the bacteria in growth medium so it can replicate. All right, so the final step, we just put the bacteria in nutrient media. So I'm going to erase up here. And so this would be our, just imagine a petri dish, right? Your nutrient media. Now I'm going to draw the bacteria much smaller now. You don't have to do this part, but so just imagine that these would be the bacteria in the nutrient media, all right? So they will replicate. Once they replicate, every bacteria will have the plasmid inside of it. All right, so all of these will have the plasmid. Let me just put blue, 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 and red. So that is how, and bacteria, they multiply quickly. So in note, like within 24 to 48 hours, you can have a lot of bacteria. And all of them will have in the plasmid with the insulin. All right. And they have their technique as well to collect the insulin. So the bacteria, it is going to make insulin. It has the insulin gene, which means it has the instruction to produce it. All right. And this is the general procedure of a gene that we are interested in. We take it out and put it in a vector, put the vector into the host. And that will now give the host the ability to do whatever it needs to. Right? So like there was tomato to survive the cold, they took a gene from the fish and put it in the tomato. So genes is a code that gives you ability to do certain things. So by putting in the insulin gene, it gives it the ability to make insulin. Right? Can I clear the screen?
Wait, sir, let me screenshot this. All right.
Yeah. All right. If this should come up and they ask you for examples of restriction enzyme, I'm going to give it the name of 3. Example of restriction enzyme. Look for the specific type of what it does and tell you quickly.
So what them word there about ind and bomb?
No, a 3. BAM is called BAM H1 and HIND3. So they are examples of restriction enzymes. So just remember the name. They make a particular type of cut. Let me just check right. Which one does this 6? Give me a second. All right, so you have a next one. It's called A3. No, these restriction enzymes, they cut the DNA in 2 ways. So for example, A3 it will cut the DNA strand, just cut it in 2, not vertically, but well, you can see it on the board, right? This is called a blunt cut. All right, so just cut the DNA in 2. All right, there's a blunt cut. You have a next cut, it's called staggered cut. So just note 2 enzymes and type of cut. They have blunt cut and they have staggered cut. With the staggered cut now, it won't go straight through the DNA strand like the blunt cut. So for like BAM H1, let me see if it was BAM H1 that did this. Now HIND3 will do this. So this is how it will cut the DNA and it leaves it with sticky ends. So we call it sticky ends. All right. So you have 2 types of cut, blunt cut, and you have staggered cuts. When you have a staggered cut, we say it leaves the DNA with sticky ends, meaning, and so you would have A bonded to T, and T, T, C, G, A. Right? So notice all of this is gone. So these ends, it would be free. All right, so we call it sticky ends. So know a few restriction enzymes, and now you know the type of blunt and staggered. Right? Right, we're going to go to next. All right, we're going to move from there now. Let's get into some variation.